Thank you, PR, and the organizers for inviting me to speak today. Also, thanks to John Pargon and Tony for giving a really good introduction to the virus and the human response. And now I will attempt to give you an overview on T-cell responses. So my favorite cells are T-cells, and I'm sure all of you know a little bit about T-cells, particularly people working in the HIV field. So the adaptive immune system can basically be separated into two arms: the humoral immunity arm, which Tony mentioned about, that is mediated mainly by B cells and antibodies that bind to pathogens or a known epitope. And the other arm is the cell-mediated arm, or the T-cell arm. And these are mainly orchestrated by two types of T cells that we know as CD4 and CD8. But there are other innate like T cells that I won't be covering today, including mate cells, gamma delta cells, and NKT cells. So these T cells basically develop and arise in the thymus, where they undergo various processing and selection, and they egress out into the periphery as naive cells. And once they encounter their cognate antigen, generally within lymph nodes or secondary lymphoid organs, they differentiate and form memory cells or effector cells that have a known function. CD8s are, I guess, a little bit more straightforward because their main function is to kill or lyse infected cells using the cytolytic granules such as perforin and granzyme. The CD4s are a little bit more complicated because they're very heterogeneous, and there's many subsets that they can differentiate into depending on the pathogen that they come across. Th1s, Th17s are more involved with viral, bacterial, parasitic infections. Th2s are involved in autoimmunity, driving IgE and kind of involved lots in asthma and other autoimmune conditions. And regulatory cells actually suppress inflammation and immunity. So they kind of basically cause this homeostatic environment where you are able to mount a response to clear virus, but then it doesn't become inflammatory. And so when you look at T cells within COVID-19 research, and if you look at the database LitCovid, which basically collates all of the data, or the literature really, within COVID-19 research, and you type in T cells, publications are about 10% of the overall COVID-19 literature published in the last 4 years. So there's quite a considerable amount of work looking at and studying these cells and show how these cells are actually really important to immunity. And the very first review that comes up here asks the question, do T cells really matter? And I'd like to argue that they do matter. Why? Because T cells are very beneficial in terms of eliminating virus-infected cells, as I described before, the CD8s and even cytotoxic CD4 T cells. They regulate a coordinated immune response. And recently, and with more work now, we know that T follicular helper cells in secondary lymphoid organs form this germinal center reaction with B cells, and they promote isotype switching of B cells, of antibodies from IgA, IgM to IgG, which is more stable, and also promote high affinity durable antibodies. And failure to control viral dissemination by the adaptive immune system can lead to severe disease. And so this schematic by Paul Moss from Birmingham shows that if you do have a delayed adaptive immune response, you have an increased morbidity and a more higher chance of having severe disease following SARS-CoV-2 infection. And so Tony spoke to you about the assays that they use to detect antibodies. And antibodies generally wane quicker than T cell responses. And you can measure T cell responses at the acute and the convalescent stage. And now more and more data is showing T cell responses following vaccination and hybrid immunity. So I'll go through in detail the three main methods that are used to detect T cell responses, and these include the ELISpot, the ICS, the AIM assay, or the activation-induced marker assay. I won't go into detail about HLA class 1 and class 2 tetramers or multimers because there's not a lot of studies out there, so difficult to compare the different results. And the reason for this is the difficulty to make HLA class 1 and in particular class 2 peptide tetramers. It takes into consideration the different HLA allele frequencies within different ethnic populations, as well as we still don't really have a full grasp of the immunodominant peptides that are required to make these tetramers. So the first assay, the ELIS spot, or enzyme-linked immunosorbent spot, so it's very similar to an ELISA, takes a few days to run. You have a plate that is coated with antibodies that bind to cytokines. Primarily, interferon gamma is the main cytokine that is detected, but now it's been adapted to look at IL-2 and TNF. You add in antigen, could be whole protein, or what a lot of people use now are peptide pools. Activate the cells overnight. They secrete, if they respond to the antigen, they secrete these cytokines, which are then bound by the antibodies. You add in a secondary antibody with biotin, and then streptavidin with a enzyme that breaks down the substrate, and it causes a color formation, which then you visualize on a ELIS spot reader as spots. And the readout is generally spot forming units per 1 million cells. And this figure on the right shows you a typical ELIS spot readout. In this study, they used negative controls, is no antigen or DMSO. They use SARS-CoV-2 structural proteins, including spike, nucleocapsid, membrane envelope, as well as non-structural accessory proteins. They also use the common cold coronavirus strains, and positive control generally is either SCB or PHA. With further technology, there's been development of this other platform called a fluorospot, and uses fluorescent antibody readout. So you can have, I guess, a more multiplex readout of your fluorospot, looking at interferon gamma, TNF alpha, or IL-2, or granzyme B at the same time. And so to compare different studies that looked at T-cell responses to SARS-CoV-2, luckily for me today, Bineke published a systematic review and meta-analysis 2 weeks ago, where they comprehensively looked at 495 data sets from 94 research articles that utilize either the ELIS spot, the ICS, or the AIM assay individually or in combination. They shortlisted 50 studies with 206 data sets that employed ELIS spot, and then included convalescents and vaccinated individuals in this data set. So with convalescents, you can see, so because it's many studies, some studies show you medians, mean, or geometric mean, and then some studies use the entire spike, or just S1, S2, or RBD. But you can see overall that there was higher responses, and this is just interferon gamma, ELIS spot, at the earlier time points of convalescence. So the higher ELIS spot forming units were within 1 to 3 months post-infection, and then it kind of drops a little bit following 3 months. And then with vaccination, the response is, the ELIS spot is higher. And so you can see the range for convalescents between 1 to 361 spot forming units per million cells. But with vaccination, and this varies with the type of vaccine as well. So Pfizer is between 8 to 460 for whole spike. AstraZeneca is between 5 and over 1,000. So quite a high ELIS spot readout for AstraZeneca. And then when you look at just S1, there's kind of like very similar. AstraZeneca has slightly higher spot forming units than Pfizer and Sinopharm. And so the next assay is the intracellular cytokine staining assay. And this is a fairly straightforward assay you can do within 6 hours. This uses PBMCs that have been separated from whole blood. You put it in a 5 ml flow tube, add in a monoclonal antibody to CD28 and 49D to help with co-stimulation of these T cells, because the short time frame, the antigen presenting cells are not able to perform that co-stimulation that's required. You also add in your antigen, give it a bit of vortex, and then between 1.5 hours to 2 hours, you add in profeldin A, which is a chemical that binds or blocks secretion of the cytokines within the Golgi and the ER. You do the readout at 6 hours. You add in your surface antibodies to, for example, there, CD3, CD4, permeabilize the cells, add in the antibodies that detect the cytokines, fix again, and then do flow cytometric readout. And this is an example of a flow cytometric dot plot readout. So you've got single cells or singlets, lymphocytes, live cells when you add in a live dead dye, and then CD4 and CD8 to differentiate the two T cell subsets. The middle panel shows you interferon gamma, sorry, IL-2, TNF, and interferon gamma expression in CD4s. And it's a percentage of, basically a percentage of cytokine positive as a percentage of total CD4s. And then the same with CD8s. And people now generally include granzyme B expression in CD8s as well. So with ICS, they analyzed 6 vaccine candidates from 9 studies, and they looked at 96 data sets. And so from the plot, it's been separated by what they were looking for, so interferon gamma, IL-2, TNF, or there's only 2 studies that look at interferon gamma and IL-2. CD4 responses are on the left and CD8 responses are on the right. You can see in general, there were higher responses for interferon gamma in CD4s in the mRNA vaccines compared to the viral vectors. And this was the same for IL-2. TNF alpha showed no real difference between mRNA or viral vector. CD8 responses are generally lower in CD4s, but the mRNAs had detectable CD8 responses for the 3 cytokines. And the medians basically are quite low because it's like 0.01% to 0.1% of CD4s, which is a limitation really for ICS because what you're detecting are very few cells, although you have to start off with quite a high number of cells. So the next assay, which is what we love, and a lot of people I think in the room have worked on this assay already, it's originally what we call the CD25 OX40 assay, and now it's been adopted around the world and called the activation induced marker or AIM assay. And this assay was pioneered in our lab by Dr. John Saunders and Tony Kelleher almost 2 decades ago. And it was basically conceived to be a very straightforward and easy assay to detect T cell responses. You can use whole blood or you can use PBMC. Whole blood gives you a higher magnitude response. You basically just add in like half the media, add in your antigen of interest to whole blood, and then at 48 hours you do your flow cytometric readout of these 2 surface proteins, CD25 and CD134 OX40. Has very low background in the unstimulated control, and John has used various different types of antigens to see if you can get a recall immune response. So TB, for example, CMV, fungi, Candida, tetanus toxoid, skin antigens, as well as flu vaccine. And he's shown that it correlates with proliferation and correlates with ICS as well. And so one of the first projects I worked on back in before 2014, during my PhD, was to really confirm that what we're seeing is a recall memory response. And I did this by sorting the CD4 T cells into memory and naive cells using CD45RA, which identify naive cells, RO, identify memory cells, put them back with antigen-presenting cells, add in antigen, and you can see only 45 RO-positive memory cells respond by upregulating these 2 surface markers. And then when we use CD62L and 45 RO to further stratify the memory into effector memory and central memory, only the memory cells respond, and there's different degrees of which memory subsets respond to which antigen. HIV is more effector memory, tetanus toxoid is more central memory. And then another talented postdoc in the lab at the time used another marker, CD39, to identify antigen-specific T regulatory cells. And we could show that by using CD39, you get a population of cells that express high FOXP3, which is a transcription factor that identifies Tregs. And we showed by looking at the methylation level of the FOXP3 promoter in these cells that they are hypermethylated, open, and actual Tregs, and we've also done functional assays to show that they suppress as well. So there's many combinations now of extracellular proteins or markers that people use to identify antigen-specific T cells. In the AIM assay, CD25, CD69, CD40 ligand, OX40, 41BB, and PD-L1. But they have different kinetics. And this is one thing that we found out when I came back to do the SARS-CoV-2 research, is that a lot of these markers have different kinetics over time. So what we use is CD25 and OX40, and it's a 48-hour readout. But to get the CD8 T cell response in the same tube, we now add 41BB at 24 hours in culture, and then we do the readout at 48 to get both CD4 and CD8 responses. And so this is Bineke again, looking at 32 studies with about 80 data sets using AIM assay, and the combination they used, or they looked at, was CD137 and OX40 in CD4s. And you can see the responses in CD4s in convalescence doesn't vary much over time. So there's similar responses early on, 1 to 3 months, all the way up to 6 months. And CD8 responses are generally lower than CD4s during convalescence. And then with vaccination, you get a slightly higher response to CD4s using the AIM assay, and again, higher in the mRNA vaccines compared to the viral vectors. And we've seen an improvement in CD8 responses as well with vaccination. And this is just showing the cumulative data, where they show that after vaccination, the ranges increase for CD8 T cell, CD4 and CD8 T cell responses using the AIM assay when you have a readout either of mean, median, or geometric mean. And so the next part of the talk, I wanted to just show you what we've done at the Kirby using the AIM assay for COVID-19 research. I'll briefly talk about 2 studies that have been published and go in a bit more detail with the 3rd study. And so this is what we normally do. We get 1 vial of PBMCs. We separate it into 3 different assays. One is a 20 to 23 parameter immunophenotyping, and we do this using the Cytek Aurora, which uses spectral flow. So now our 5-laser Cytek Aurora can actually go up to about 42 parameters. We do an OX40 assay, and then we do a 6-day proliferation assay as well by using cell trace violet to find proliferating cells. We use peptide pools of spike, RBD, and nucleic capsid. And then this is just a graphic to show you the different markers and fluorochromes that we use for phenotyping, AIM assay, and cell trace violet proliferation assay. So the first kind of study that we published, and this was in collaboration with Harry and Rowena Bull, using the COSIN study, we showed that CD4 T cell responses at 1 month can predict durable memory B cell responses at 1 year post-infection. And this also correlates with the increased neutralization breadth, particularly CD4 T cell responses to RBD. And we think because the CD4 T cell responses within RBD potentially contain T follicular helper cells. And we did use CXCR5 as a marker for circulating T follicular helper cells. The next study, we want to have a look at the difference in immune response between children and adults of the same household during acute and convalescence. We did a suite of different assays, including next generation single-cell transcriptomics. Just to show you that the AIM assay, it was quite interesting at the time, was we saw no difference or no increase in memory CD4 T cells in the children during convalescence, but there was an increase in magnitude in the adults when we looked at responses to RBD and whole spike. And we think because the children were able to clear the virus very quickly, the innate immune system was able to do this. They were not able to robustly enhance a memory T cell response. And so we sequenced their TCR. It's very diverse, but their memory pool was also diverse, and it wasn't clonal as much as the adults. And this highlights the need to vaccinate children, because even though they are asymptomatic and clear the virus very quickly, they're not able to mount a proper memory T cell response. And so the third study, which I'll go into detail, because we basically looked at T-cell responses longitudinally from convalescence to post-vaccination, and this was published at the end of 2022, a big collaborative effort between us and the Garden. And early on, which is between June to August 2020, John Saunders had a look at these patients from the ADAPT study, and he used whole RBD protein from Daniel Chris' lab in the Garden and showed that convalescents, people in convalescence had higher responses to RBD compared to unexposed healthy donors. And then we used flu as a comparison antigen. He showed that there was positive correlation between the RBD CD4 response and proliferation. And then he stratified the two groups with either proliferating CD4s or not proliferating CD4s. The group that had proliferating CD4s had higher IgG/IgM score and also higher neutralization titer. So then when I came back to join the Kirby in about September 2020, I really was interested in this initial finding, and then we wanted to tease this out in further detail. So we had 12 patients from ADAPT that we defined as being antibody high, and these were defined as having a neutralizing titer greater than 40, and 12 individuals with that were termed antibody low, and these people had neutralizing antibody titers of equal to or less than 40. And you can see even in the antibody high group, their neutralization titer decreases over time. So at 8 months, you see a significant drop in the neutralization titer, and it goes with the general theory that antibody does wane over time. And we showed that the antibody high group had good neutralization breadth to beta, gamma, and delta, and this also decreases over time. So figure C is 3 months and D is 8 months post-infection. And so our readout, CD8 is at the top, and you can see CD69 versus 137, 25 versus OX40 134. Generally, CD8s do have a higher background, so when we background subtract, and we look at spike, RBD, or nucleocapsid responses, what we found was the group with antibody high had much higher spike-specific responses in CD4s at 3 and 8 months, and also responses to RBD, and not so much to nucleocapsid. And we saw no difference in the CD8 response. And then when we looked at proliferation, this was the same trend again. We saw higher proliferation in CD4s that responded to spike and RBD, and nothing besides nucleocapsid for CD8 T cells. And following vaccination, it was able to rescue the RBD-specific response in the antibody low group. So these people were vaccinated between 12 and 8 months post-infection, and you can see there was a significant increase in responses, CD4 responses to RBD. Again, no real difference in CD8 responses. And this correlates with the increase in neutralization titer after 2 doses of vaccination, either AstraZeneca or Pfizer, for antibody titers to ancestral and delta. And when we correlate RBD-specific CD4 responses to neutralization titer, we saw a positive correlation. So this highlights the importance of vaccination, particularly in individuals with low immunity during natural infection. We can't assume that everybody's going to get the same level of immunity once they've been infected. And so to tease out those antigen-specific CD4 T cells out in more detail, we utilized single-cell transcriptomics, the 5' kit, including TCR, or T-cell receptor sequencing, on the 10x genomics platform. We also did bulk TCR sequencing of the proliferated cells to see, basically, when we overlay the 2 data, whether the same clones that we saw in the OX40 AIM assay also proliferate. And so this is an example of a patient. We did 4 of the antibody high group. We sorted spike-specific, RBD-specific, and nucleocapsid-specific cells in the Q2 quadrant. We also sorted the unstimulated cells using 45RO ex vivo memory cells as a control population. And this schematic shows you the 10x genomics platform. So use a microfluidic droplet, oil droplet platform. You have one B that's coated with a DNA barcode. You put one cell into the same droplet, lyse the cells, or the mRNA contents bind to the barcodes on the beads because they have poly-DT. And then you basically do a series of PCRs and form libraries, and then sequence using the Illumina platform. And so Wenhua Ku from the Garden did all of this transcriptomic bioinformatics for me. And we showed with the ex vivo memory, we can find, we identified 7 populations, 3 populations that had a cytotoxic signature, which is quite interesting, 2 with T regulatory T cells, and this, I have to highlight, is actually underestimated and actually not detected at all if you use CD137 and OX40 or CD40 ligand. And so CD25 OX40 in combination identifies antigen-specific Tregs. And we also had 3 populations of memory, CD4 T cells, CXCR5 central memory, which are most likely T follicular helper cells, activated central memory, and transitional memory. And we annotated these subsets using known genes that are highly expressed in these subsets. And this is just showing the proportion of the different subsets within the antigen-specific cells to nucleocapsid spike and RBD. So there is quite a high frequency of antigen-specific Tregs that people don't really report, together with the cytotoxic Th1-like cells and the T follicular helper memory cells. And we also looked at the TCR of these antigen-specific clones, and all of this work was done by Catherine Jackson at the Garden. And we showed that there was high TCR diversity in the antigen-specific populations compared to the ex vivo memory. We were able, hasn't come up, but we were able to show there was usage of diverse TCR alpha and beta chains. What you should see are ribbons. I think it's a MAC to PC difference. And the main TCR beta that was used was 21 and 7.4, and alpha was 2.2. And what you should see here are connecting lines, but it's disappeared. And then when we got the TCR sequences from the proliferating cells, we overlaid this onto the UMAP from the AIM assay. And what we showed was the proliferating clones were actually majority cytotoxic, with some CXCR5 central memory and Tregs for both for nucleocapsid spike and RBD. So contrary to a lot of literature, Tregs do proliferate, and cytotoxic CD4 T cells are there and mounting an immune response. And so this is just again to highlight that with the CD25 OX40 AIM assay, we detect proliferating CD4 T cells. And so there's a quest, I think, for like a pan T cell assay. And so Bineke tried to address this by comparing the 3 different assays. So they had 58 donors with hybrid immunity who were naturally infected, then vaccinated. They used the same samples, did AIM assay, ICS, and ELISpot, and they used spike-pooled peptides. And this is a really nice study because it showed with CD4 AIM, the CD25 OX40 gives you a higher magnitude of response or frequency compared to CD137 OX40. CD69 and 137 is higher than 25 and 69. Gamma for ICS is generally higher than the others, the other cytokines. And the fluorospot was no real difference between the 3 higher granzyme B. And you can see that there's polyfunctionality when you look at the cytokines in combination in ICS. And so what assays should you choose? And I think then we have to go look at the limitations, because both ELISpot and ICS assays is associated with implicit a priori assumptions about how important interferon gamma is. And I think that now we know it's a very heterogeneous population of cells, so we can't just focus on one cytokine. It doesn't take into account the other cell responses, and most often only measure Th1 and Th2 responses. The ELISpot can only measure a couple of parameters at a time, and then you have to either deplete or purify, separate the different CD4, CD8 subsets to determine where the cytokines are coming from. Otherwise, you won't be able to tell. and you cannot do live isolation of these cells for downstream functional or molecular analysis. The AIM assay, however, can detect more global population and higher levels of antigen-specific T-cell clones than ICS and ELISpot, and it allows live isolation of these cells for downstream functional and molecular characterization. And Bineke did a correlarogram to show you how all of the assays correlate with each other. And they found that the OX40 CD25 AIM assay correlate very well with ICS and interferon gamma ELISpot. The CD8 AIM assays don't correlate too well, but I think it's probably because the peptides that are used are 15-mer peptides, which are more presented by HLA class 2 and not class 1. And so the CD8 responses are much lower in magnitude when detected. And then to summarize all what we know about T-cell responses to SARS-CoV-2, vaccine-driven immunity, when you do head-to-head comparisons of the different platforms, the mRNA platform induces the strongest CD4 and CD8 response compared to viral vector platforms. Protein adjuvanted or inactivated vaccines do induce appreciable CD4 responses, but are less effective in inducing CD8 responses. And in immunocompromised individuals, they can respond to COVID-19 vaccination, but to different degrees. And alterations in B-cell compartments correspond to decrease in humoral and antibody response. But in general, CD4 and CD8 T-cell responses are maintained and conserved. And we've also done a little bit of this in immunocompromised cohorts as well, and that's what we observed. And several studies that showed vaccination and booster of immunocompromised individuals are associated with significant protection. And so when you talk about durability of T-cell responses, they are durable, and regardless of disease severity, they can be detected in asymptomatic infection. Slightly lower durability observed T-cell responses to vaccines compared to natural infection, but hybrid immunity increases T-cell responses, shown by groups in Karolinska and in the US, maintenance of conserved epitopes and enhanced recognition of mutated epitopes. And Paul Moss showed that he was able to trace T-cells, or the half-life of T-cells, out to 200 days. But now with reinfection and vaccination, the maintenance of these antigen-specific or SARS-CoV-2 specific T-cell pools are maintained over time. And I think the response to SARS-CoV-2 is supported by T-cell memory responses that have been able to be detected in people that were infected with SARS-CoV-1 18 years after infection. And so T-cells, memory T-cells are still around and able to mount a good immune response when it encounters the virus. And so I'd like to thank everybody involved in the studies that I presented, and thank you again for the invitation.